some things that from your mTOR C1 or C2 lecture you remember, and some things that we're going to go over again today. So, so let's just take a look here at the beginning of our MAP kinase signaling pathway, which is important for you to know. It's either important for you to know right up until the end of the day on December 2nd, or it might be important for you to know for your career development if you're going to be at all interested in items that are going to be likely to treat cancer or treat other things that the MAP kinase pathway is involved in. So, we in this case are starting with a receptor tyrosine kinase, which has the ability to, once it's dimerized, phosphorylate itself. It's phosphorylating the other half of the dimer, and it phosphorylates on tyrosines, hence the name tyrosine kinase. And it's a receptor because it binds to a ligand on the outside of the cell. And once that tyrosine gets phosphorylated, it can interact with sites that have a binding site that will recognize phosphotyrosine. And what would that binding site be called? Let's hear it nice and loud. What on a protein binds to phosphotyrosines? What domain binds to a phosphotyrosine? SH2. Let's all say that. SH2 binds to tyrosine phosphates. Okay, good. Let's keep that in mind, because the other half here, it has an SH3, which stands for sarcomology. SH2 binds to phosphotyrosines. The SH3 on the other side of this little molecule called Rab, what does that interact with? It interacts with RAS, and that's what this is, and it's got some prolines on it. SH3 binds to high proline domains. So this is where that's going to bind on this site. Now, this is not RAS. This is the son of 7-less, which is actually going to activate RAS. So if it activates RAS, what is it going to do to this little RAS that's binding here in the plasma membrane? What is this binding to, the RAS itself? GDP. And it's a GTP ACE. So you've got to get the GDP off, right? So what is this doing? It's exchanging the guanine nucleotide from GDP, allowing GTP to bind. So we call that a guanine nucleotide exchange factor, or a GEF. So that's how this whole process gets started, because then we go from RAS. Now you remember the funny name, how it got to be SOS, and now we come to, now we've got this RAS that's been activated. Now we start a whole pathway that has 3 kinases, one in a row. And the first one is going to be called RAF, and RAF is going to be sequestered in the cytosol, bound to a protein called 13-3-3, which tends to bind a lot of different proteins and keeps them occupied in the cytosol until they can be called to the membrane protein that they need to interact with. So the way this works is that this protein now binds to RAF based on a regulatory site, but in addition it's transported over there because it's bound to phosphotyrosines over here. So these kinases have phosphorylated the RAF for it to bind to the 14-3-3, but it gets reconfigured as the 14-3-3 drops off, and now we have phosphorylation at other sites. So this now becomes a kinase that can be activated once it gets phosphorylated itself, and it activates a MAP kinase, ERK kinase, or a MAP-ERK kinase, at its serines or threonines, and then it phosphorylates in dual positions. One is a tyrosine, and the next one is a threonine. So that activates our MAP kinase, which then can interact with a dimer itself, and that allows it to get into the nuclear pores and cross into the nucleus if it's going to activate transcription. So out in the cytosol, it's going to activate a 90,000 molecular weight protein that's called regulator of signaling kinase, RSK, and that comes into the past the nuclear pores and can now phosphorylate this signal response factor that binds to the polymerase complex. And the MAP kinase itself is phosphorylating, what's this called? Transcription control factor, and that, when it gets phosphorylated, can also bind to the polymerase complex, and it binds via this serum response element. So then our transcription can take place. So we don't have that big polymerase complex bound on here, but just imagine that it is, as this regulates this response element so that we can initiate transcription. So that's a review, essentially, of our tyrosine kinase receptor pathways that can take us all the way from the receptor, where it interacts with itself to form a dimer on the plasma membrane, all the way to where the MAP kinase can initiate these phosphorylation series to get our serum response element activated. So, so we talked a little bit about that, and then we got to the point where we were going to talk about some kinases that were very similar, except that the ability to phosphorylate itself was not entirely present on those dimers. And so that's a family that we're calling the cytokine receptors, because most of the ligands that are going to be binding here are binding to the, are going to be cytokines that are going to be binding to this extracellular surface. But very much like the tyrosine kinases, they dimerize once they get ligand binding, and this now allows, in this case, a new protein to come to the surface and do the actual phosphorylations. So whereas the tyrosine kinases are going to be binding to a growth factor, and in this case we're seeing this EGF. This is what's going to be dimerizing, and you can see that there's really not so very much on the inside of the cell membrane as there is on the outside. So these are fairly large domains for binding the growth factors themselves, which are going to be coming from other proteins that have been proteolyzed from plasma membranes. So all of this is taking place on outside of cells. And you can see that now our ligands are not the little itty bitty ligands that we thought about with our G protein coupled receptors. Most of those that you've talked about so far have been small hormones, but this is almost a large protein itself, and allows the two domains that are the binding domains to clamp down on this protein, and now it's part of the complex. That opens up this region for dimerization. So what we're seeing is a dimerization that can then allow the two intracellular domains to interact with each other in the tyrosine kinases. Now, keeping that in mind, what we have for our cytokine receptors is very similar. Here we have dimerization, ligand on this side, which is completely parallel to the dimerization that you see for the receptor tyrosine kinases. And here we have a ligand binding site that's going to be binding cytokines. And once that happens, then we get what would be our dimerization, and that's based on the interaction with that large protein ligand. And when that happens, we call in a kinase coming from the cytosol. And in this particular case, it's called the Janus kinase, and is often abbreviated JAK, Janus kinase, and we call that JAK. So JAK is going to be binding here to this membrane surface, and it is the kinase that then does the phosphorylations. So this phosphorylation lip now becomes open once the kinase itself is activated. Bless you. And at this point now, we can see that that the JAK kinase is going to begin interacting with a substrate, and that substrate is going to be called STAT. So here we have an example. This is erythropoietin, and it's binding to the EPO receptor, and the particular kinase that it's coupling to is JAK number 2. So does anybody know what erythropoietin does? Yeah. That's what? Yes. So what it does is it takes your progenitor cells, erythroid progenitor cells, and these are the EPO receptors demonstrated all along this surface that are going to respond to the hematopoietic stimulus here, our erythropoietin, and then it will begin to make a whole series of new cells that will ultimately mature into full red blood cells. So it's going to be a mitogen, and it's going to be activating the production of proliferation of new cells. And you can see in a developing embryo, there's a region here that is responding to this. This is where the EPO receptors are, and you can see that this is where our JAK kinase is accumulating as well. So we essentially have our erythropoietin binding to its receptor. It regulates JAK. JAK can now interact with multiple kinds of proteins. So the first one that I told you about was a protein called STAT, which gets phosphorylated by this Janus kinase, and that leads to transcriptional activation. So we can get new proteins being made, and those are going to be including those that are necessary to move along in a cell cycle to begin to develop the proliferation of the cells themselves. But it also binds to GRAB. Now, where have you seen GRAB in the last 10 minutes? Okay, your short-term memory needs to start kicking in here. So GRAB was that protein that we saw that was binding to the phosphotyrosine on the tyrosine kinase receptors based on its SH2 domain. It's a little protein that has SH2 binding sites on one side that can bind to phosphotyrosines. On the other side, it had an SH3 that can bind to phosphoprolines. And here you're seeing it again. So this is a multifunctional little coupling protein. It's only there to be able to bind to other things. And in this case, what's it binding to? RAS. So, so it's performing that very same function based on JAK phosphorylation that it did before. So the JAK is phosphorylating that cytosine, that erythropoietin receptor, and now it can bind to GRAB. It can interact with RAS, and it can begin that entire MAP kinase pathway. So that isn't really so so very different from the receptor tyrosine kinases, except that it required JAK to be bound in order to get the phosphorylation. Now here you see a phospholipase C, and this is phospholipase C gamma. So you remember from our lecture number 2 from me, what did we talk about the phospholipase C is doing? Yes, IP3 production. So it takes the inositol phosphates that have already been phosphorylated on their 4 and 5 position, and it cleaves them. So now you take the phosphate head group off, and that goes along with it. So you have IP3, very water soluble, can get into the cytosol, and finds its way to the endoplasmic reticulum, where it has an ion channel as its receptor. And what does that ion channel let out into the cytosol? Calcium. So now your calcium is there to do multiple things like transcription activation or repression, modification of other cellular proteins. How's it doing that? How does free calcium do all of these things? Calmodulin. So calmodulin has its own kinase that it interacts with, right? Or PKC. So the other thing that was left in the plasma membrane after you took the IP3 off was diacylglycerol, right? So the PKC gets activated by the calcium and then gets further activated at the plasma membrane by binding to the diacylglycerol. And so that's not going to help us much with our transcription, but it's going to cause what we're calling here modification of other cellular proteins. That modification is phosphorylation. So mostly this calcium is going to work that way. Okay, and here's our PI3 kinase. What does that do? It phosphorylates PI3 at the 3 position. So it's adding a phosphorylation to the sites that are on the inositol. So if you already had 4 and 5 phosphorylated, now you've got the 3 phosphorylated. What is the significance of having these different hydroxyls on the inositol phosphorylated?
They can signal like certain proteins.
Exactly. So for those proteins to be able to be bound to the plasma membrane and do functional activities at the plasma membrane, this is the key. They come and bind to these inositol phosphates, and they've recognized the pattern based on charge distribution. And so what was the name of the binding site that would bind to these inositol multiphosphates? What did we call that binding site? Most of these binding sites are homologous to something that was the very first protein it was discovered in, as a clue. So there's lots of proteins called homology proteins. So that SARC, SH2 is SARC homology 2. SH3 is SARC homology 3 because they happen to be related to the SRC kinase. In this case, the protein was plextrin. So it's the plextrin homology domain, and people abbreviate that PH. There are just so many abbreviations, and people babble on and on and on with these abbreviations, and then they don't even remember what they were there for. So TCF. Who remembers what that is? It was on the previous slide. What? A bank. Okay. Well, that's one way of remembering it. And in fact, when the convention center in Raleigh, it was the RBC Center, which is actually for Royal Bank of Canada, I think. Royal Bank of Canada. But of course, for a biologist, every time I drove by there and saw the RBC Center, I thought, oh, blood banking. So anyway, PI3 kinase, very important because it can call in another kinase called protein kinase B. And so we now have the ability to, again, modify cellular proteins, transcription activation. So all of these can be activated in response to that JAK that binds to that site. And here's a picture of what it looks like. Here's our erythropoietin right in the middle of the dimer of these two monomers of the erythropoietin receptor. So again, erythropoietin is a very large protein interacting with the two proteins that are the monomers in order to pull them together and allow the JAK to ultimately bind to the C-terminal region on the inside. So here's a schema of another JAK-STAT pathway, and this is in response to leptin. Now who knows what leptin is? It's a satiety hormone. So what that means is it is the receptor in your brain that is allegedly telling you that you've already had plenty to eat, you don't need to eat anymore. Allegedly. I think it doesn't work very well. Because I do think that the reward pathway trumps anything that responds to the energy detectors that are going to be in the hypothalamus. So, you know, that Krispy Kreme, you're going to want that. In fact, I go to Thanksgiving dinner with my daughter's in-laws, and they have this routine. After Thanksgiving dinner, they walk down to Krispy Kreme's. Now, why do you need a Krispy Kreme after you've just had not only the entire Thanksgiving dinner, but the pumpkin pie?
Why wouldn't you?
So leptin is supposed to be responding to the fact that your stomach is engorged, you now have insulin running through your bloodstream, you've got CCK coming into the brain, you have all those signals that are telling leptin to tell you to stop eating. And what do? We all walk down to Krispy Kreme's because it's a family tradition. So anyway, leptin, this is its receptor. It's going to be dimerizing. It is going to be interacting with the JAK, and in this case, again, it's JAK2. Responds to that stimulus by phosphorylating itself, and phosphorylates all these tyrosines over here on the intracellular region of the leptin receptor. So now you can call in other proteins like STAT, and that binds here to this phosphotyrosine. And then the STAT gets phosphorylated by JAK. So now the STAT isn't going to be staying there very long. Once it gets phosphorylated itself, then that takes up this region where it would have been phosphorylating or binding to the leptin receptor complex. And so now it can dimerize itself. So two phosphorylated STATs will dimerize. Now that opens up the nuclear pore signal so that it can pass into the nucleus and serve as a transcription factor. So a whole variety of new proteins can then be made in response to this. So this can set up a whole pathway. Here's our cytokine JAK-STAT pathway. Again, this is all from the Science signal transduction series, if you want to take a look at some of those. So our receptor calls in JAK, gets phosphorylated, it phosphorylates STAT, and STAT dimerizes, and we get new gene expression. Now, one thing I want to point out to you is the way that we can stop all of these things, and that's from what does this PTP stand for? Pardon? It's a phosphatase. So this is protein tyrosine phosphatase. So it is going to be taking these phosphates off the tyrosines so that you can stop this response ultimately. So once it gets the fact that there's phosphotyrosines on, it's going to bind to the JAK, and it's going to bind to this receptor, and it's going to take those phosphates off. And it can also reverse the STAT pathway. Okay, so we've got our dimerization here. We've got our phosphorylation along the intracellular surface, so we can call in new proteins. Some of those are going to be substrates for further phosphorylation themselves. And once they get interacting with each other, you can see they're binding to their phosphotyrosines. So what kind of a binding site are they probably going to have? An SH2 domain. So on their little sites on STAT, we've got an SH2 domain so that it can interact with its partner. How terribly convenient. So this SH2 is going to interact with the phosphotyrosine that's over here on the other side of its partner. So isn't it nice? You can get a dimerization here. So that gives us the opportunity to get phosphorylation. Now here's how we get rid of it. Here we have the ability of our, let me see here, a protein phosphatase called SHIP. And this is going to be binding to a phosphotyrosine. So that's the first clue that it has a job to do. It has to get rid of these phosphates. So the first thing it does is bind to one of them. So it binds to one of them, and that actually opens up what would be, so what a lot of phosphatases and kinases is, and they have an auto-activation site. And what that means is part of their molecule has now bent over and covered over the actual catalytic site of the kinase, or in this case, the phosphatase. So that you can see this is covering what would be the phosphatase that would be active. So while it's in the cytosol, it's simply not active because it's covered over its own site. Once it binds to this phosphotyrosine, it sort of rips this open, and now it allows the catalytic site to remove this phosphate from the JAK itself. So JAK is no longer going to be able to phosphorylate the receptor or other things that get bound to the receptor. So this is a very good way of now eliminating our phosphotyrosine continuing on in this pathway. Now, the other thing that we can do that is much more long-term is to be able to remove this by a pathway called the ubiquitin pathway. So ubiquitin is the signal that now takes the proteins that need to be removed to a proteasome, and just as the name says, that is an accumulation of proteins that are going to be proteases, that are going to start breaking down the proteins that have been marked for degradation. So the marking for degradation begins again with phosphotyrosines. So at this point, we've got our phosphotyrosines, and there is a protein called an E3 ubiquitin ligase that is in the cytosol that's going to recognize these phosphotyrosines, and it is going to ligate ubiquitin to that site. So now the ubiquitin can be bound to these sites, and that is the signal now for these sites to now be recruited so that they can be degraded. Now, these little sites, first of all, they have an SH2 domain. So that's the domain by which they can bind to the phosphotyrosines. And then they have two other little domains that are important. One is called the complex of SKP1, Cullen, and F-box. And so again, these are named after the proteins that they were first discovered on. And the abbreviation is the SCF domain, and this makes this an SCF protein. So it's that is going to be allowing this to recruit the ubiquitin ligase. And that's going to put the ubiquitins on these proteins. So does anybody have any questions about that before we go on to something just a tad different, but actually fairly similar? Okay, so we've talked about how we get phosphorylation and how we remove it. And if you go back and take a look at your mTOR lectures again, you'll see some of these proteins popping up in those complexes of either mTOR C1 or mTOR C2. One of them, we've seen ERK as a phosphorylation. We've seen some other things. Now I'd like to go into the NF-kappa B signaling pathway, which is actually regulated by the breakdown of a suppressor or an inhibitor. And so when I say this is entirely different, it's not going to be involving the kinds of mechanisms that we just talked about, but it is going to get to the point where the signal is going to be based on a phosphorylation. So this is the general schema, and here we have interleukin-1 receptors that are going to be binding with a TNF-alpha receptor so that we have a complex. Now you could probably say, well, that's a little bit like our cytokine receptors, except those were homodimers. And this is two entirely different proteins binding to two entirely different things. Now another thing that we might have in common is that once they now have gotten together based on their ligand binding, then they interact with a kinase, which is going to phosphorylate a small kinase called I-kappa B. It's going to phosphorylate this inhibitor of the kappa B pathway. Now this just happens to be an alpha and a beta and a gamma. That has nothing to do with the alpha, beta, gamma G proteins. That's not what we're referring to. This is the I-kappa B kinase. Now this can also be regulated by some other things that we don't know exactly how they work. So physical and chemical changes both can interact ultimately to get this kinase activated. Now this is a serine 3' kinase, so that's a little bit different from the tyrosine, but what it does is it interacts with a protein that's the inhibitor of kappa B. So this alpha subunit, once it gets phosphorylated, now it sequesters what would be our NF-kappa B. So the NF-kappa B is two proteins of two different molecular weights. One is 65,000, the other one is 50. And while it has this inhibitor bound to it in the cytosol, it cannot act as a transcription factor. So it's pretty much curtailed in its activity. But once this gets phosphorylated, now what we're doing is calling in this E3 ligase to these phosphorylated sites that were in response to this kinase. And that now makes it possible to degrade the inhibitor. So once this gets ubiquitinated, and these little circles represent little ubiquitin molecules, so it usually gets ubiquitinated more than once so that you have a series of little ubiquitin molecules here that signal. And so that takes this inhibitor protein to the proteasomes for degradation. Well, that frees up the NF-kappa B. So the way it does that is that these sites were hiding nuclear localization signal. And once the I-kappa B has been removed for degradation, then that opens this up, and this little dimer, this heterodimer, can now come into the nucleus and become a transcription factor. So all of the genes that were responding to TNF-alpha or interleukin-2, and most of these are considered to be pro-inflammatory. So the kinds of proteins that are going to be now produced here are going to be the kinds of proteins that are involved in the very first phase of the inflammatory response. So they're the pro-inflammatory proteins that are going to be induced. So that includes some very important things. So it includes iNOS. We talked about iNOS already. So iNOS, that's the inducible nitric oxide synthase. So that's going to be induced in cells that produce a great deal of nitric oxide for the purpose of killing cells or bacteria. Cyclooxygenase 2. You all know what COX-2 is? So these kinds of, it makes prostaglandins. So your non-steroidal anti-inflammatory drugs, your NSAIDs, all work by blocking. But COX-2 has some very special blockers that have been fraught with some untoward side effects that have to do with cardiovascular system. So some have been taken off the market and some haven't. But COX-2 is induced at this very same time. So the prostaglandins and thromboxanes can be made. Phospholipase A2, that breaks down phospholipids and releases the arachidonic acid that's usually at the 2 position of the phospholipids, so that it provides the substrates for cyclooxygenases. And then it's got some adhesion molecules, and interestingly enough, it now begins producing some other cytokines like interleukin-1, so that can provide a source of continuing this, and TNF-alpha itself. So your whole pro-inflammatory response can actually get magnified as this whole pathway comes through. So it's very, very convenient. But interestingly enough, and I don't know why I don't have that on here, one of the things that it does induce is the I-kappa-B alpha subunit again. So while you have removed it here as part of this initiation step, one of the things that this is now producing is the very protein that needs to come back in order to curtail the response. So it will induce that along with all these other things, so that it will have a protein to inhibit any further activity of NF-kappa-B on these cells. Okay, and I wanted to just go over how some of this can be involved in toll-like receptors. The Nobel Prize was won 2 or 3 years ago by 3 people from throughout the world who were all involved in toll-like receptors. So these are not looking for an individual cytokine or an individual protein. These receptors are very general, broad-based receptors that are looking at some very important things that are not specific, like lipopolysaccharide. So that means that any microorganism that has lipopolysaccharide on its surface is going to trigger a response of toll-like receptor number 4. Or things like short-stranded RNAs. So that's the way that a lot of organisms are going to curtail the activity of a host cell that they've invaded, is to make a bunch of short RNAs that can then cause the breakdown of your larger single-stranded RNAs. So it recognizes just this kind of a motif. Flagellin. So So it's going to recognize parasites that invade. And this just happens to be in the brain, but this is all over. So these are the receptors that really respond in the immune system before your B cells and T cells can begin to come into play. This is the native, basic, general purpose immune response that can occur before you begin to get the more regulated immune responses. And most of these are going to be setting up that same kind of pathway that we just talked about. So you can see here's a cell that has toll-like receptor 4 responding to lipopolysaccharides, toll-like receptors 1 and 2 responding to lipoproteins, and these others. And they essentially get to the point where they are going through a pathway that can phosphorylate our ICAPA inhibitor so that we can now begin to remove that inhibitor, and our NF-kappa B can enter the cells and begin producing TNF-alpha, interleukin-1, and initiate that same immune response without having to go through a cytokine stimulation itself. So those are the pathways that are left from that previous talk. And if you'd all like to stand up and stretch for a minute, I am going to take this down. No, that was not.
That's what I've been doing.
Yeah, that's what I was saying.
Also doing that before the paragraphs in a row.
That's just like the extra credit. Like every extra credit assignment, I did the same exact thing.
Or I could just like, do you know how many points the first again was? Well, Taylor's
I don't know. I just assumed you did
Would you need-
100. That's actually a really good question.
I want to thank you all for sending me your homeworks. I was just wondering what I was going to do when that turkey was cooking. So perhaps when everybody else goes to get Krispy Kremes, I may just pull out my iPad and send you all some messages over Christmas. So.
Okay.
An egg ray? Oh,
For
okay.
Christmas.
She needs
You don't
to
care about any
know.
comments that we might have? Yes.
Is there any way I can make the outline here tomorrow, please?
Yes, please.
Very-
Oh.
That's a lot.
I thought it was Tuesday night. Oh, you're right.
You put it as today.
It was, yes, it was Monday, right? Sure.
Oh,
That
you
would be-
are awesome.
Thank you.
Even though I'm gonna be on the road tomorrow, so I
I
gotta do
still
it
got to
today.
I'll take some of the pressure off.
Well, it takes a little pressure off of me too, because then I can get your other set. Review.
It's just one day. You can move it to
So,
next week.
so we have a very similar homework assignment that is, Steve, is that posted already
Not yet.
for next week? Okay. Which would be due on the 30th. So how long do you want?
Longer
Because-
than the 30th.
Right.
Yeah.
Over Thanksgiving break?
No.
Wait, I'm sorry.
You make it do on test day.
Yes.
What's the
Yes.
assignment?
Today's assignment is to put the outlines for the part of the reading that was on our reading assignment list that you haven't read yet. But I put that as due on the 30th.
So it's just
So the 30th is 2 days before your exam. So that would probably give me a little chance to get them all back to you. You know, I can get them back to you after you have sent them to me. if you-
And that's one
Yeah.
that Steve's gonna post in a little bit.
Yes.
Okay.
So it's not, this is due tomorrow, all these three.
was due tomorrow, but I'm up to negotiation here.
Yes.
And
So you're saying just
unfortunately,
make them
I
all
don't
due
charge
next
money,
week?
so we can.
So make them all due next week.
Well, I think that beyond Thursday might be a little bit hard, but I could probably say Friday morning,
Okay.
this Friday morning.
For this one, for the first
This
one.
one, the one that was due.
Yeah.
Will give us.
But that at least gives me Wednesday. I'm gonna try to do it today, but like I don't want the pressure of having to do it
No.
today.
The assignment for today would be due on the 30th. So the assignment that
How
was
was
from
she
last
talking to?
week, that was
It's
supposed to be
actually not
due
bad.
today, if you wanna have it until Friday, that would be okay with me. Friday morning. Anytime during the day on Friday. So if you're visiting relatives,
I'm like reading
really
textbooks
out
off
in
the
the country,
computer,
they don't
so I'm gonna have to print all this out.
use Wi-Fi and you have to find a Starbucks, that'll give you a chance to do that.
Because I have to scan this in, so I need to
Um,
finish it today.
I don't know about you, but my sister
You
has
know
a
you can use
rotary
your phone.
You know what those are?
Especially if she's writing so small
That's
on
a
a pencil.
You just don't know how to take good
Yes, and then
pictures.
Stephen will post.
Cause my scans are
Yes,
legit.
yes.
Like I did.
Or when I
on
get
a
up to my
scan.
office, I can email it.
Look
Okay,
at my
now
balance
I wanna talk to you a little bit about
they're
small
very
G proteins.
Oh my gosh, she looked like
And, there are multiple families, but, if you can just remember about what those families are and some generalizations, that would be fine. But the two families I'm gonna talk about today are so these are all RAS-like proteins. And it's easy to pinpoint them when you're reading along in a paper, because most of them are named with something that is 3 letters as an abbreviation, and the first one is an R. And then they get numbered. So you get a whole series of numbers of things that have roughly the same name. So we're going to talk a little bit about the RAB family proteins. See, RAB begins with an R, 3 letters. So, and then the ARF. Now there's an example of something that doesn't fit into that. Because ARF stands for ADP ribosylating factor. And it was discovered many years, actually, before small G proteins were discovered, as a small factor, and nobody knows what it is when they call it a factor. So they're kind of cautious. They don't call it ADP ribosylating protein if they're not sure that it's really a protein. So this was named factor, ADP ribosylating factor, and it's now a whole family of G proteins. So these are very similar to what you've already seen. This is our RAS. It's got a GDP bound to it, and when the GDP is removed, then you can get a GTP bound. And this GPP NHP is simply an analog of GTP that cannot be broken down or hydrolyzed because it doesn't have that oxygen here that's the substrate. So because they don't have the regulatory portion of the heterotrimeric alpha subunit, they're generally smaller. So they're about 20,000 molecular weight. And they don't have the beta gamma subunit. And they never will because they don't have the ability to interact with it. So when GDP is bound, it's inactive, just like our heterotrimeric G protein. And the RAS protein was initially discovered as a GTPase. It was an oncogene of sarcoma viruses, actually in chickens. And so, so that was why the RAS was found to be very important, is that it was found that in many mammalian tumors, that this had a mutation. And what that mutation did was it reduced this GTPase activity that would have been taking place automatically, as this is a GTPase, so that it never went back to the inactive state. So it made it continually active as if it was going to be activated. Now we know now that since that is part of this MAP kinase pathway, which is a mitogen, that this is what causes continual growth. So these are the families that we have here. We have a host of different kinds of analogs of RAS that cause cell growth and proliferation and sometimes differentiation. We have one called Rho, and that largely interacts with active cytoskeleton, but can also be involved in cell proliferation. The one I'm going to talk about today mostly is the Rab family, and they're involved in vesicle trafficking. So getting proteins that are on vesicles from hither to yon, anywhere in the cell, is under the direction of a specific Rab protein. And so these are the addresses on the envelope. RAN, these are involving transport between the nucleus and the cytosol. ARF proteins, ADP ribosylating proteins, these activate phospholipase D, and I'm going to talk to you a little bit about that in the next half hour or two. And then a few other things that can be involved in various things like ion channel regulation, cell shape, cytoskeleton. So we've got hundreds of these, and here is their family tree. So the way these family trees work is that you have some mysterious stem progenitor from archae periods of time, and then as they move into something that's going to be a little bit different and diverge, then you see the divergence here. And the space in between really represents how long ago the divergence took place. So things that are very close to each other up here, like row A, row B, and row C, they diverged relatively recently in phylogenetic time, from each other. But they diverged very long time ago from the family that turns out to be the Rab family. So you see these diverged from their families all at these intersections that were quite some time ago. So this one is the Rho family, which includes Rho, RAC, and cyclin-dependent protein number 42,000 molecular weight. So these are here, lots of Rho subtypes, and other proteins that are fairly similar. Some are in yeast, some are in mammals. Our RAS family is the family that's over here. You see lots of RAS names. Our ARF family is over here, and there are ARF-like proteins called ARL, ARF-like proteins, AR as in ARF, and L as in like. And the RAM family here, and the whole RAB family is here, although the RAB family is absolutely huge and is much bigger than what's represented here. So this is a list, and you can see RAB family is lots of RABs, and they're all given numbers. So here's our RAS family. It includes some other things like RAP, RAL. You can see this motif. And one that's very important called REB, which deviates because it's got 4 characters here, R-A-G-B. And if you look very carefully on your diagrams of your TOR-C1 and TOR-C2 pathways, you're going to find REB is involved in there. So go back and take a look at that and see what that is. Now, this is how this works. We've got our GTPase activity is going to break down what would be the GTP and remove the gamma phosphate and inactivate our RAS. It's got a little switch on here that holds this in place, and you need to have a GEF, guanine nucleotide exchange factor, in order to open this up and allow the GDP to be released. So you can see this opening up and allowing the GDP to be released. You can pay some attention to that. And then that allows GTP to bind, and then this closes back up again. So the reason this is an oncogene is that the GTPase activity that's taking place up in here won't take this phosphate off. So it becomes what we constitutively active. Now I'd like to talk about the Rab family. And the Rab family, here I'm labeling all of these. These are GTPases also, so they all work the same way. And I should also tell you that in order to get these GTPases to work, you sometimes need to have a GTPase activating protein, or a GAP. So most of these small G proteins are going to have a GEF specifically for them and a GAP. And they might have great specificity. Not one GTP activating protein is going to be useful for virtually all of these small G proteins. So just think of the host of different kinds of proteins now that we have. We've got not only all of these G proteins, but we've got GEFs, GEFs, and we've got GAPs, GTPase activating proteins, for each of these. So that's now thousands of proteins. And you need to think of them in terms of broad generalizations, because they're going to be in different kinds of cell types for different kinds of purposes. So in this case, all the cells, this is very ubiquitous, have these Rab proteins because they all perform the same kind of functions in these cells. So these functions are generally of two general categories. One is involved in exocytosis, and that's the process of taking a vesicle that is carrying cargo inside it and releasing it out. So all of your hormones that are going to be made and stored in vesicles, like insulin, this is going to be regulating its release. All of your neurotransmitters are perhaps going to have some Rab proteins that get involved in that. And the second one is vesicle transport anywhere. So we can start out with endoplasmic reticulum going to Golgi, trafficking from the Golgi to the plasma membrane or any other organelle, and trafficking to return from a plasma membrane and endocytosis. So here's a general schema of a cell. And here's our nucleus, and here's our endoplasmic reticulum. And you can see that some things, like number 1 and number 2, are responding to interface little vesicles that bud off from the endoplasmic reticulum and need to go to the Golgi, and that would be Rab number 1. Or little vesicles that bud off from the Golgi and need to go back to the endoplasmic reticulum, and that's Rab number 2. So that's what these little circles with the numbers mean, is these are all a small G protein. So think of them as the address label. So the address label says here, leave the endoplasmic reticulum and find your way over here to the Golgi. So the proteins are going to have little motifs on them that are going to be recognizing some kind of a receptor on the Golgi that can recognize them and bind that vesicle there. So now we need to get our proteins that are on vesicles to bud off from the Golgi in this trans-Golgi network interface and find their way to either a storage vesicle or to a plasma membrane. So here, for example, is GLUT4. That's our glucose transporter that responds to insulin. And the way insulin works after you've gone through that whole series of MAP kinases is it takes what would be a vesicle that contains glucose transporters just sitting in adipocytes or skeletal muscle waiting to move. And these 10, 8, and 4 are going to move this vesicle very rapidly to the plasma membrane, where it can merge, and then you've got hundreds of new transporters almost instantaneously, as long as it takes to get through that tyrosine kinase pathway, because these are already here. So they were transported here by these one of these 3 Rabs. Melanosomes, kind of specialized tissue, again, that needs to have a color change in an animal that needs to go from white to black very quickly so that they're not going to be eaten by a predator. So again, this is a vesicle that's here, and it's Rab number 27 that moves melanosomes specifically. Now we also have things that need to go to some integrins, our Wnt pathway that we'll talk about next tomorrow. These are going to be responding to what would be vesicles that may have been internalized. So our internalization, we've got lots of Rabs that may be involved, and generally we want to think in terms of the early endosomes, the very first internalized vesicle, as having a Rab5 on it. And then that Rab5 can take it to late endosomes, and here we've got another Rab7, that's going to take it to lysosomal vesicles. So, so just to go over this a little bit here. Why is this not moving? Oh, okay. Our pathway of vesicle internalization for G protein coupled receptors is very keenly associated with desensitization. So here's our G protein coupled receptor. The Nobel Prize winner, Dr. Lefkowitz, would call it a 7-transmembrane receptor because he doesn't think the G proteins are as important as binding the arrestin. So once this gets phosphorylated with a GRK, or a G protein coupled receptor kinase, that phosphorylation calls in arrestin. And arrestin has a site on it for binding to clathrin. So clathrin is the structural protein that begins to cause an invagination in our otherwise smooth surface of the plasma membrane, so that we now get what's called a clathrin-coated pit. So these little dotted lines represent that clathrin as it begins to coat the pit. And then ultimately, as soon as we get lots of clathrin forming here, and in case you're interested in how this works, these are what they call triskelions. It's a little 3-pronged protein that binds to itself other 3-pronged proteins. And if you go home and take a look at your soccer balls, you'll see that as these form triskelions, they form 6-membered rings, and then the 6-membered rings condense to a 5-membered ring, and that takes up less space and causes this to become more like a ball. So if you look at your soccer ball, you'll see it's got 5-membered and 6-membered rings. That's how a soccer ball is made. And that's exactly the way that this is made, is those 6-membered rings become the 5-membered rings, and that causes the curvature. So then it gets budded off with a little protein called dynamine. So once it gets into the cell, it gets uncoated by an ATPase that removes all this clathrin, and that's where you see little other vesicles that have Rab5 on them binding to this site. So the Rab5 is going to begin in the formation here and then be the hallmark of an endocytic vesicle. Now these endocytic vesicles then have to move and fuse with an early endosome, and that's what the Rab5 does. Now if it goes anyplace else, it needs another Rab. So the Rab5 has to leave and bud off so it can go back to do other trafficking of other vesicles, and you get a Rab4 binding here, which can make it now bud off into a recycling vesicle that can then take the protein back to the plasma membrane. So to look at that in a little bit greater detail, here's our Rab5. It's located beginning on the plasma membrane because the little Rab5 vesicles can bud off and go join this as it begins to internalize. But then it is the hallmark of the early endosomes. This directs their trafficking so that they can join either recycling endosomes, or they might bud off and go to the Golgi, or they might form these multivesicular bodies and ultimately find their way to the late endosomes. But it's going to take a different Rab to do each one of those steps. So the first Rab after the early endosome could be the recycling endosome. So this early endosome could actually stay in the cytosol for quite some time if it never gets a RAB4. And that could be very useful because if this is going to be transporting along with it the whole MAP kinase pathway, and you'd like your MAP kinase to be continuing to phosphorylate for a fairly long time, you want to have sustained internalized vesicles. So we have a whole family of vesicles that are endosomal that are not going to recycle very quickly. But as soon as it gets a RAB4, this is the rapid recycling back to the plasma membrane, where it can either remove cargo. So, for example, if it's taken in via its transferrin receptors, if it's brought in a transferrin, you want to remove that transferrin and let it go back out so that it can bind iron. So it can act as an exosome. Okay, so RAB4 is going to be taking it back to recycle. On the other hand, you might want it to be transported so that it actually gets broken down and removed altogether. And this is one way in which we can have what we call down-regulation of receptors. So those receptors that may be internalized could be desensitized because they're no longer interacting with G proteins. But on the other hand, if they get recycled back to the plasma membrane, then you haven't changed the number of receptors. But the way you change the number of receptors is to take them out of this early endosome through a multivesicular body and transport them down to a late endosome, which merges with a lysosome. So the RAB7's job is to do that. So we want to take things to the lysosome if we're going to have them degraded. Our endosome here interacts with RAB7. Once that vesicle has now got the RAB7, now it can interact with a receptor site for the RAB7 on the lysosome called HOPS. And once that has bound, then you can begin to get some fusion through fusin proteins. Now, if we want to regulate recycling of our endosomes, we have some signals here that are important for binding another RAB called RAB11. And these are the slow recycling. So these are the ones that are likely to be continuing to signal through the beta-arrestins that are going to be on their surface. And they'll continue to signal right up until the RAB11 takes them back. So whereas the RAB4 was our rapid recycling, the RAB11 is binding that maintains it here for a while and then gradually sends it back. Now, one of the things that can happen while it's internalized is that any of those phosphate groups that were put on when it was initially desensitized from a GERD, the phosphatases are in the cytosol, and that's where they come into play, and they can remove those phosphate groups so that when you take these receptors back to the membrane, they are dephosphorylated, and they're like native receptors all over again. So this RAB11 pathway can be a very important one. So you can see lots of different address labels that are going to really describe how that early endosome manages the receptor numbers on the cell surface. Okay, now as you can imagine, there must be some really important diseases, and so here's another slide set that you can stare at about things that are those diseases that may be involving dysfunctional RAB proteins. And since we've got over 60 of them, and their numbering is not, they're in chronological order of when they were discovered. So they don't have numbers in some logical way about what's going on in the cell. But here's a list of some of those diseases, and you might find something useful. So I highlighted this tuberous sclerosis complex, TSC, which interacts with RAB5. Where have you heard of TSC in the last 5 days? Well, you might go back again and look at your TOR C1 and TOR C2s and see if you can see something that looks like TSC on there. So, some things that might be important to know, RAB proteins in the immune response and infection. So there's RAB13 that's, when it's dysfunctional, tends to be associated with Crohn's disease. Dysfunctional RAB5 can disrupt interactions that are important in the immune response. So it can cause infections. Some of our lipid trafficking. So here's RAB5, RAB4, because this internalization process is extremely important for getting the lipids transported and brought back into cells for storage. So HDLs and LDLs all bind to receptors that are part of the pathway here that in order to get them into the cell, get the lipids removed in the lysosome and distributed to the cell, you have to have these RAB5 and you have to have RAB4 in order to take those receptors back again to the surface. And that has to occur very rapidly. So this turnover of receptors is taking place within 5 or 10 minutes. Whereas with the G protein coupled receptors for hormones, that may take minutes to hours. Yeah.
With a lot of these RABs being pretty disease specific, as well as a variety of physiological things, how easy is it to target a specific RAB, and do you then see a lot of side effects because of the wide range of physiological functions we do?
So the question was, with this myriad of different G proteins, how do you target or how does the cell target?
Well, how do you, because I
Like, do
mean,
people
it's
target
Okay.
drugs?
Yeah.
So probably the best way would be using an antibody that's specific, because most of our polyclonal antibodies are made by an epitope that's a fairly short sequence from the protein, so they can be very specific. So it could be an epitope that may be 5 amino acid sequence and maybe fairly specific for one RAB versus another. So that's predominantly how people would recognize them. Then our studies, once you've known the sequence, then you can either knock it down or overexpress it. So most of what you're going to see in research literature is knocking down or overexpressing things. And that's where you begin to see where dysfunctions are in the cell pathways that you might be interested in.
But in people, isn't it pretty hard to do a knockdown or overexpression?
Yes, so in people, probably what's happened is the cells that are not working very well, if the pathway has to do with lipid trafficking, then you have to look at what some of the components are in the lipid
And
trafficking.
downstream more so than
Yeah.
the RABs themselves.
Yeah.
Got it.
Some cancer, RABs. Okay, so I want to just switch gears, talk about the Rho family and the ARF family for just a minute. So Rho, RAC, and CDC42 are all generally involved in cytoskeletal actin regulation. So under the plasma membrane of all cells that have any kind of structure whatsoever is a series of actin filaments. So these are actin dimers that then polymerize into a filamentous or fibrous structure that forms the structure part of a cell. If the cell is going to move or the cell is going to grow in any particular direction, it has to break down that structure and then reform it. So let's just say you were a macrophage and you needed to move now to a site of an infection. Then that structure that you you already had built up is going to have to be dissolved. So the filaments are going to have to be broken down into dimers and then rebuilt again so that your front end can begin moving forward and your rear end has to catch up. So that's how a macrophage is going to move. So it's got to have the breakdown and the reformation. The breakdown is under the influence of Rho, and the reformation is under the influence of RAC. So these proteins are going to be involved in breaking down that actin and then reforming it in its new structure as the cell moves forward. So that's generally what those are, but I'm going to show you a couple of figures that show that Rho can also interact with phospholipids, and it does that by activating a phospholipase D. Now you saw phospholipase C removes the head group and takes the phosphate with it. Phospholipase D removes the head group but leaves the phosphate on the phospholipid. So now it is phosphatidic acid that you have left on the plasma membrane, not diacylglycerol. So diacylglycerol has no charge. Phosphatidic acid is an acid. So it's going to be damaging to the plasma membrane and kind of act like a detergent because one end is going to be charged and the other end is going to be the two fatty acid side chains. So that's exactly what a detergent is. A detergent is something that can bind lipids on one end and bind water on the other end. And that's what phosphatidic acid does. So it's like creating a detergent right there in the plasma membrane. So that can be very helpful if you want to change your plasma membrane size and shape and move a cell forward, because it can help you reconfigure that plasma membrane itself. So that's involving that. The other family we're going to talk about is ARF, because that also regulates phospholipase D. So for the next 5 minutes, we're going to take a little look at some data. And so I decided to give you the actual data, so we can pass those around. So this is where those figures are, and we've also included the method.
Are they all the same?
Yeah, it's just one page. Okay, so I've run out already, so you guys just keep passing them back until the guys in the back. Okay, so this is the ARF family and one row. We'll take a look at this. So the method is to be able to look at the phospholipase D activity. And phospholipase D, since it's breaking down, that's a hydrolysis step, right? So an enzyme like phospholipase D is going to need to have water, which is going to be breaking that bond between the phosphate and, in this case, it's going to be phosphatidylcholine. So, so what you can do if you want to assay this is get something radioactive, but not water, to substitute for the water in this breakdown. And so frequently that's going to be radioactive ethanol, but in this case it's butanol. So now you've got phosphatidylbutanol because the radioactivity is on the site that stays with the phosphatidic acid. Okay? So that little assay is up at the very top of your sheet here on page 1. So that's the assay for these two figures that we're going to look at. Now, it's going to be causing an effect that's going to be breaking down, and so what we're going to see is this breakdown of our phospholipids. And this is a cell type, it's a human astrocytoma. And it has lots of receptors for lots of G protein coupled hormones. And some of them, thrombin and thromboxane, are up here. radioactivity is going down, the more of this toxin you have in there called Bruffeldin A. So Bruffeldin A, it turns out, is an inhibitor of the small GTPase ARF. So you can see that some of these hormones, and in this case it's the H1 histamine, the B2 bradykinin, and the M3 muscarinic receptors, are all very sensitive to Bruffeldin A in their production of this phospholipase D activity. So this is enzymatic activity, phospholipase D, breaking down the phosphatidylcholanes. So what does that tell you about these two groups of hormones and their hormone receptors? Knowing that Bruffeldin A will block ARF. Yes, so for these, ARF must not be involved because it can't be, the phospholipase D activity is not reduced. But for these, it gives you the idea that ARF must be really critical to that phospholipase D activity. And it's not going to be phospholipase C because that doesn't leave the phosphate there to be the receptor for this radioactive butanol. So what we have to assume now is that ARF must be working with muscarinic receptors, histamine receptors, and bradykinin receptors to produce a cell signal. And that signal is going through the phospholipase D. So does everybody agree that that's probably what this is telling us? Whereas these must be working through something else. Oh, but they still can have like a phospholipase D activity, but it's not going to go through ARF. Okay, so here we've got now the question of how does this work? This is the muscarinic receptor assays over here. This is the thrombin. So keep in mind, over here, thrombin did not require ARF. It was not blocked by Bruffeldin A. So that's over here. So now the question is, how can we piece out using pharmacological blockers as to how this is working? So the first blocker is abbreviated U73. It's a drug company product, so it's got a long code name, but this is a phospholipase C inhibitor. So what was the question being asked? That's this here and this here. Right, the question is this phospholipase D activity based on something that goes through phospholipase C? And so for our muscarinic receptor, is this different from this? No. For our thrombin receptor, is this different from this? So can we assume that phospholipase C was the instigator of the thrombin response? Okay.
So thrombin needs both phospholipase D, or works through phospholipase D and C.
That's what you could be led to believe, because again, this is this very same assay. Now it could be that once that phosphate is there to make phosphatidic acid, there now comes along another enzyme that takes the phosphate off to diacylglycerol. Okay? So that's phospholipase D. So it can work either through phosphatidic acid as a substrate, or it could look at the whole phospholipid. Either way, it's going to be able to work through this assay. So yes, After phospholipase C, then phospholipase D could come along. But this does require phospholipase C first. So you could assume then that this is probably working, since it's G-protein coupled receptors, probably working through what kind of a heterotrimeric G protein that could activate phospholipase C? Q, GQ. Or how about phospholipase C gamma? What was activating that? Phospholipase C beta is the one that's activated by GQ. Yes, the pathway that takes us through the tyrosine kinase pathway is also going to produce the activation of a phospholipase C. This time it's gamma. Okay, now the next question, pertussis toxin. What question is being asked there?
Is it involved in receptors, or does it activate receptors?
Okay, so what does pertussis toxin block?
GIO.
GI and GO. So the question would be, is GI and GO involved? So, is it involved in the muscarinic response? No. Is it involved in the thrombin response? No. Okay, so it's not a GI-GO pathway. Now we've got a Rho inhibitor, and it's called C3 exotoxin. They took out the word botulinum. Maybe they didn't want to scare people. Because I used this before when I was at another university, and when they sent around, the safety office sent around a list of things that you have in your lab, and as soon as I put C3 botulinum exotoxin, oh my God, all H broke through, because the safety office came, they got the state involved, where was I storing this, what was it? Well, it turns out the exotoxin is not what kills you. So if all I had was the exotoxin, it was not necessarily a safety issue, but the word botulinum was very frightening. So this is it's called a Rho inhibitor, C3 exotoxin. Is it involved here?
Yes.
Yes. Is it involved with thrombin? No. So we must infer now that the Rho family must be involved in this as a small G protein. So now we can take and add, using genetic overexpression systems, either more Rho, we can use a wild type, or we can use a dominant negative. So here's our wild type. When we add more Rho, we get, what happens? We get an actual increase in activity, as opposed to something that blocks Rho, which gave us a decrease. Did anything happen over here with the wild type adding wild type Rho? Nothing. Okay, so Rho's not involved here at all. When we put in our dominant negative, what do we have? A decrease. What happened over here? Nothing. Okay. So one last figure. How do we regulate this? So our Rho inhibitors, we've got all of these. So we can conclude here that we've got phospholipase D activated. It can be activated by a PKC in response to some hormones, or it can be activated by ARF or Rho by others. So it can do the small G protein or the large heterotrimeric G protein. Ultimately, this would be activating, moving from phosphatidylcholine to phosphatidic acid, which can act as a detergent. So this can be involved in how the cells and the vesicles fuse, because you need a detergent if you're going to get vesicle fusion. So we've just created our detergent. It's such a nifty little thing. So how do we get this activated? Here's mu opioid receptors. They're involved in endocytosis, and they regulate a phospholipase D, and this is one with ARF. So again, you can go back and take a look at this. Take a look at what happens with your Burfeldin A compared to controls here once you've added DAMGO, which is the activator of the mu opioid receptor. this is entirely reduced by adding brifildin A. So you know that mu opioid receptors must have to go through this as well. Alpha adrenergic receptors, here's ARF6, and again, a very similar kind of scheme. So take a look at these figures and see as you understand what's going on. Here's our internalization in response to an alpha adrenergic stimulator, and here's what happens as the receptors begin to get returned to the plasma membrane. So those are our regulators, ARF and Rho, that are involved in this. Oh, this was the slide I wanted to get to. So they need to be activated. ARF needs to be activated. If we had a G protein coupled receptor, what is the guanine nucleotide exchange factor for a heterotrimeric G protein? No. It is so obvious. What causes the release of the GDP on a heterotrimeric G protein? Agonist binding to the receptor. The G protein coupled receptor is the GAF. So, and Tyler, you can tell us about GAPs, right? No. So, what for a heterotrimeric G protein is your lab studying, which causes a change in the GTPase activity? There's a whole family of them. They begin with an R. Regulators of G protein, RGS.
Oh.
So, yes, so there's a whole family of RGS regulators of G protein signaling, and they're the gaps for heterotrimeric G proteins. But here you can see we've got a protein inhibitor for the gap called RNO, and we've got a protein inhibitor for the gap called GIT. And so what you can see here is when we have our ARF guanine nucleotide exchange, we can see that this is a little protein, this is the GEF, and that stimulates these effects. And so that's here. And our GIT is our GTPase activating protein, and that inhibits the internalization. So we've got GIT is this one down here, and the squares are the RNO. So you can see by modifying the activity of our ARF that we can see changes in our internalization. So this is going to be stimulated by the beta-arrestin. So you can see we can begin to get this interacting now with what we've known before. So go back and take a look at these. The only ones that are on this page that I just gave you are just the first couple that we've looked at, but that gives you a chance to look at what the methods were. And so we can, you can go back and take a look at some of these other figures and think about them and think about how the ARF is working through these. So tomorrow we'll continue on with